Alrighty, hi everyone. It looks like we are officially live and streaming, and hello. It's so great to see everyone joining this live stream. It is 11 o'clock on the dot over here in Santa Cruz, California, but we are going to give folks about another minute or two to join our live stream, get settled in, and get excited for today's program. So hello and welcome everyone. It's so great to see all of you. Alrighty, and as we are waiting, please feel free to go ahead and say hi in the YouTube live chat. Tell us where you're watching from, where you're tuning in from. We'd love to see Seymour Center supporters all over California and even the United States too. So go ahead and say hi in the chat. Alrighty, so it looks like we are 1 minute past the hour over here, so we really don't have any more time to waste. So let's just go ahead and dive right in and get started. So hi everyone, and welcome to an exciting Labside Chat with the Seymour Marine Discovery Center. My name is Jen, and I will be your host for today. If you have seen a Labside Chat before, welcome back. If this is your first time tuning in for a Labside Chat, we're so happy that you're here. To help set the stage and to tell you a little bit more about what you'll see today, is that today's program gives us the opportunity to have a Q&A style discussion with a scientist from UC Santa Cruz and discuss how scientific research plays a role in the understanding and conservation of the world's oceans. We have a great program lined up today because we have the pleasure of talking with and asking questions to Dr. Haley Ohms. Dr. Haley Ohms is an assistant project scientist at UC Santa Cruz and a researcher with the Fisheries Collaborative Program. Haley, thank you so much for joining us this morning.
It's my pleasure.
Awesome. Well, I'm really looking forward to asking you lots of questions and learning way more about your research. But before we dive in, I do have a couple of housekeeping items right up top that I first want to address before we start getting to all of those juicy questions. So first off, Haley and I want to acknowledge the tragedy that happened in San Jose. We want to extend our thoughts and hearts to our neighbors just over the hill from us in Santa Cruz. So it's a sincere tragedy, and we are thinking about you. Next off, I want to give a big shout out to UC Santa Cruz's Fisheries Collaborative Program in partnership with NOAA Southwest Fisheries Science Center for sponsoring today's chat. We are so grateful for your support. Additionally, thanks to everyone who submitted questions in advance for Haley that I will most definitely be asking during our conversation today. Y'all are so awesome because you submitted so many questions, and I hope we get to all of them during our short time together. However, if you didn't get to submit a question in advance for Haley, not to worry at all. You are always more than welcome to submit a question in the YouTube live chat so we can see it in real time. Alrighty, so housekeeping is done. House has been kept. So Haley, I say let's just dive right into those questions. Alrighty, so to kick us off, Haley, can you tell us what it means to be an assistant project scientist at UC Santa Cruz or a researcher with the Fisheries Collaborative Program?
Sure. I guess it it means that I'm a scientist. I conduct research primarily on fisheries, and I focus all of my time on that research. So a lot of people at the university will also be involved in teaching. But in the Fisheries Collaborative Program, our primary focus is on research.
Yeah, so that's cool. So you are really focused on research, and you said that you work on fisheries. So I'm assuming you have to be working with fish, right?
That's right.
So can you tell us what type of fish you are working with?
Yeah, so kind of throughout my career, I've worked on this group of fishes called the salmonids, which includes salmon, trout, and chars. But right now I'm working on steelhead and rainbow trout that, and primarily in the Carmel River, so very specific to California. Yeah, so steelhead and rainbow trout are my primary focus right now.
Nice, nice. And so steelhead and rainbow trout, I think I've heard this before, is that they are the same species but have different common names. So can you help clear that up for me, please? Because somehow I am still getting confused about, are they the same? Are they different? If they are different, what is the difference?
Yeah, fair enough. That's a great question. So, steelhead are anadromous, meaning they are born in streams. They migrate out to the ocean, like you would think of a salmon doing, and then they migrate back to the streams to mate or spawn, we call it, and reproduce. Rainbow trout spend their whole lives in streams. They don't migrate to the ocean. Steelhead are much bigger as a result of their ocean migration, and rainbow trout are quite small. So, like 4 to 6 inches, they can be quite small. They, steelhead and rainbow trout belong to the same species. They mate with one another. So despite their size differences and their behavioral differences, they mate with one another and they produce offspring that can either be steelhead or rainbow trout. So the two of them are combined in a species and in populations.
Mm-hmm. Gotcha. Okay, great. Thank you for clearing that up. I know that I always get confused on that topic, so great. And you say that when you're working with steelhead and or rainbow trout, what exactly are you looking for with these fish? Are you measuring them? Are you counting them? Are you looking at swimming patterns, migration routes? Are you putting tracking devices in them? Like, what's going on? Like, what are you doing with these species?
Yeah, I guess all of the above, really. So, I mean, kind of the shorthand is what I say is I study the ecology and evolution of them. So, like broadly, I'm looking at the way that they interact with the environment, what their population dynamics are, and sort of why they do the things they do, why they have certain behaviors and migratory patterns and things like that. So, right, I spend a lot of time counting fish, measuring them. My work in the Carmel involves a lot of tagging fish with pit tags. So we're using these small tags that are about the size of like a large grain of rice. We put tags in a bunch of fish and then we monitor where they go, the time at which they move, and also how they survive.
Yeah. Oh, that's so cool. Okay. So before we dive in, I start asking very specific questions about what types of research is going on with pit tags, et cetera. But you keep referring to the Carmel River, right? So that is your study site, it sounds like, where you're really working with steelhead and rainbow trout. So can you tell us a little bit more about where Carmel River is located in the world and like what type of ecosystem it is?
Yeah, so the Carmel River is on the central coast of California. So it's just north of Big Sur and south of Monterey Bay. Um, and it is like what is it? It's a Mediterranean climate ecosystem. So think like oak savanna kind of landscape, really dry in the summer, sometimes really wet in the winter, sometimes not, depending on the year. Um, yeah.
Yeah, great. And so we kind of like have this picture in our mind about what Carmel River looks like, where it is in the world. And so now we're really focusing on these steelhead and where they're going and how they're migrating and how big they're getting and whatnot. So you were talking about pit tags, which sound really, really interesting. And you say that it's what, about the size of grain of rice, goes into a fish. We're able to see where it goes. So can you tell us a little bit more about that technology and how we're able to track the fish and like what you were able to learn from your tracking studies?
Yeah, so pit tag technology is the same. The pit tags are the same kind of tag that you'll put in a dog or a cat to microchip your pet. It's also the same technology that's used in stores to prevent theft. Right, when you have an item and you walk out of the store and there's those two kind of like gray things on the side of the door, that's an antenna. And what's on your item is a little tag. And so when you walk through, the antenna detects that. It's the same thing with fish. We put these little tags into them, and then we set up antennas throughout the river um, that record when a fish moves through. So we can look at our data logger and say, oh, fish 127 moved through on May 5th at 2 p.m. And what's really great about that is that the tags are small, so they don't have a battery inside of them, so they last for the full life of the fish. We can tag a fish when it's less than 1 year old and still be detecting it, 5 or even 10 years later. It also allows us to get a lot of information, basically to spy on the fish. I mean, this is like a lot of what I do is just spying on what they're doing. When are they moving? Why are they moving? At what time are they moving? That sort of thing.
Yeah. And so what is your theory about why they're moving? Like, what's going on?
Yeah, well, steelhead and rainbow trout like to use different parts of the watershed at different parts of their life. So, in the Carmel River and in a lot of Central Coast streams, there are some fish that rear, so they're living and growing in the watershed, and they stay there, and then they migrate out to the ocean about 2 years after they were born. Other individuals will migrate down to the coastal lagoons within the first or sometimes even in the second year of their life. Those coastal lagoons are a really unique thing to the Central Coast, or I guess coastal California streams. And so we're using Pittags to really understand when and how often those movements occur, and also how important those habitats are within the watershed.
Yeah. And so these pit tags are that we're using to figure out where these fish are going. Are we able to also figure out where the fish are going in the ocean, or are they only just used in the freshwater part of their life stages?
Yeah, that's a great question. They can only be used in freshwater because they work on radio frequencies, and those waves don't travel well in saltwater. So they're really only used in freshwater. I mean, the benefit is that when a fish comes back as an adult spawner, we get to detect them again. But you're right, we don't get to use that technology in the ocean. People are using things like acoustic tags or satellite tags to look at ocean movements.
Yeah, understood. So when you have a steelhead and it grows in the A stream and then eventually migrates out to the ocean and hangs out for a little bit of time, does it come back to that same stream to reproduce? You touched on this a little bit, so I was hoping that maybe you could help me understand a little bit more about this life cycle that it goes through.
Yeah, that's a great question. We think that most fish, for example, that leave the Carmel River and migrate out to the ocean, most of those come back to Carmel. That they come back to their natal stream, is what we call it. Not all of them do, and we call those strays. And we think that that's actually a really important adaptive feature of the species. That if one population locally goes extinct, these strays from other populations can move in and recolonize that habitat. But yeah, primarily we think of them coming back to their natal stream. There are some really interesting exceptions, and in fact, there's some great work coming out of Scott Creek, the Scott Creek group within the Fisheries Collaborative Program, where they've tagged juvenile fish in Scott Creek and other places, and seen them even as juveniles moving across the Monterey Bay. And so there's always some oddballs, right? But by and large, they seem to come back to their natal stream.
Gotcha. And like, how is the steelhead population doing in Carmel River? It seems to be popping up in the news that, it's not doing great, it's doing okay. There's different things happening in the river. So what's your general assessment of the population?
Yeah, steelhead in the Carmel and really coastwide, I would say, we're not seeing big declines, but we're also not seeing huge rebounds either, right? We're not seeing like big population increases. And so a lot of my work is focusing on why that is. In Carmel, there's been a lot of restoration. So the San Clemente Dam was removed, but there's been a lot of other restoration by the water district and the water company in Carmel. So yeah, I guess to answer your question, they seem to be kind of holding steady.
Yeah. And why do you think that is, that we're not seeing any increase in population growth?
Yeah, I don't, that's the
That's what
$1 million
you're
question, right? That is the $1 million question. We're working on figuring out why that is, right? And what's complicated about this species and the, you know, a lot of salmonids is that they spend time in the ocean, right? And so we're trying to figure out what is limiting them in freshwater, but then we also have to be looking in the ocean and, you know, trying to figure out what's happening with a fish in the ocean. I mean, just sort of that is, you know, a metaphor in itself, right? It's hard. I think some of the things that I point to in terms of populations not rebounding are, you know, continued high water use, right? There's a lot of people in California and there's a limited amount of water. And so I think that continues to be a hindrance.
Yeah, definitely, definitely. And so I'm just so grateful that we have folks like you working on this question to help give us these answers. So we're just so grateful for your work, and it's so fascinating to see what you're finding out. So that's it's just really, really great news to have you on this problem. So you were talking about the San Clemente Dam. And so I was hoping that we could back up a little bit and talk about this dam, what it is, when it was installed in the river, when it was taken out, and how the steelhead population is responding to its removal.
Yeah. So the San Clemente Dam was a dam located kind of right in the middle of the watershed in the Carmel River. It was built in the 50s, if I recall, and it was removed in 2016. So it was a really exciting event in the Carmel. And the main conservation goals of removing that dam were to improve fish passage and to improve sediment transport within the watershed. So fish were able to move upstream and downstream of the dam via a ladder and some other kind of assistance, but it definitely hindered fish movement. And dams also trap sediment behind them, right? If you imagine a river is flowing downstream, it's carrying sediment with it. Steelhead and rainbow trout use gravels to build nests and lay their eggs in, and they're very picky about their gravel. They have a certain type of gravel that they use, and so, you know, those gravels are being trapped or were being trapped behind San Clemente Dam. So since the dam removal, what we're seeing is fish movement, both of juveniles and adults, through the project area. We're seeing juveniles and rainbow trout using what used to be reservoir now is stream habitat. They're using that for rearing, so they've gained a little bit of stream, and we're seeing a lot of sediment movement. And so in those three ways, the dam removal was really a success.
That's really great news and really great to hear. And so, but you briefly mentioned a ladder of the dam. What is that? Are fish actually using a real ladder to like cross a dam? Like what's going on there?
Yeah, that's a great question. So, right. There are these things called fish ladders, which they come in a variety of styles. But if you imagine kind of like a trough with these flow checks to kind of slow down the water, there's like water flowing through this trough, and the fish are able to swim up and jump into each of these little troughs and make their way up and around a dam.
Okay.
Or up and around any sort of barrier. They're really commonly used to move adult, you know, steelhead and salmon upstream.
Yeah, gotcha. Oh, okay, great. That makes a lot more sense.
Yeah.
So, this dam removal, you know, it's part of this restoration project. And so you briefly talked about different restoration happening along the Carmel River over many years. And so, in addition to removing dams, like what other restoration techniques or tools or management actions can help to promote population growth of steelhead?
Yeah. You know, the main one that I've been thinking about a lot lately is lagoon management. So this again is something that's unique on the central coast of California. And when I talk about lagoons, you know, if you go to the Santa Cruz boardwalk and you walk across the Trestle Bridge, you see the San Lorenzo coming down into the ocean. And in the summertime, there's a big sandbar that blocks the river from entering the ocean, right? And so all of the water pools into that, what is a lagoon, we call it. Those are really productive habitats for juvenile fish, for juvenile steelhead, but also coho and other salmon in California. Um, we humans really like to tinker with those lagoons, right? Because there's important property that borders them. And so we drain them and, we do a lot of things to manage the water levels there, and for good reason. But we are also at the same time learning a lot more about how important they are for these juvenile fish, and that there's some indications that these can really be a bottleneck for these populations. And what I mean by that is if we improved lagoon function, made them a little bit more, fish friendly, that we could see really big positive impacts for the population. At least those are the early indications of some of our research. And so when I think about management actions, I mean, that's one that I'm starting to think about. Like, are there ways to manage these lagoons in a more fish friendly manner? What are those ways? And I think it's possible. I think that some of the kind of water management techniques that we're thinking about are very doable. And so I'm actually quite hopeful that that's something that will come about in the next, I don't know, 5 to 20 years or something.
Yeah, well, I'm excited to stay up to date with your research and just continuously learn what you're finding out. It sounds fascinating. So, alrighty. So we got a question here asking if you have ever encountered issues with light pollution on steelhead or rainbow trout. And if you have seen any type of effect on this species, whether it be negative, positive, no effect maybe, in case if light pollution has any negative effect on the species.
That's a great question. I haven't looked, and I to be honest, I haven't thought about it. So the honest answer is I don't know. I think it's a great question, and I actually think that light pollution is kind of an understudied and underappreciated effect on a lot of species. So it's a great question, but and unfortunately, I don't have a great answer.
Hey, that's totally fine. I think that is what you said, is that, science is always advancing. We're always learning new things, and hopefully we'll learn more about that topic in the next couple of years. So, alrighty. And so, Haley, I'm so curious, like, did you always want to work with steelhead or rainbow trout or these migratory fish? Is this something that you always were super passionate about?
Gosh, I think yes and no, right? I think I grew up fishing with my family. My dad was a commercial fisherman, and I've always really enjoyed working with fish. When I was an undergrad, I had the opportunity to go work in Bristol Bay to count salmon. So Bristol Bay is in I grew up in Alaska. Bristol Bay is in Southwest Alaska. It's the biggest sockeye salmon fishery in the world. And the way that they manage that fishery is in part by having people count fish as they move into the river. And so then they can tell commercial fishermen when to fish or when to not fish. I took that job very naively, thinking it sounded like fun. And it just, I was sold. After like a month in the field, I was fully on board for a career in fisheries. So I guess it's an interest that started fairly early on for me.
Oh, that's awesome. It just goes to show like how important these early experiences are to try everything. And then that can really, light a fire under you and can lead you in a certain direction in your career. It's very, very cool.
Yeah, it's funny. I just didn't even know that was a career, right? I mean, at the time, I just didn't even know that was an option. And so I am really thankful that opportunity came up, because I mean, at the time I was like, this is a job. How is this is a job? Okay, great.
Totally. And so can you tell me a little bit more about like your life as a fisheries scientist or as a researcher? Like, how much of your time is spent in the field working with the species, counting steelhead or putting in pit tags, or how much of your time is spent in the lab, like crunching numbers or analyzing data?
Yeah, it kind of depends on the year, and it certainly depends on the season. I spend a lot of time here where you're looking at my computer, writing, doing data analysis. That's a really time-consuming part of the job. This year I will spend most of September and maybe part of October out in the field capturing fish with a crew from the Monterey Peninsula Water Management District, and we'll be tagging fish to monitor their movements and survival. And then throughout the year, I'm out checking antennas and just kind of like keeping tabs on equipment, maybe once or twice a month. So it's a good mix. It's a good mix.
Yeah, good. And so working with fisheries, there's a lot of talk about the ecological, cultural, economic aspects of fisheries. And so can you just, I know this is a big question, but briefly say like why these three different things are so important to the fishery? Like what can a fishery bring to a cultural aspect, or what can a fishery bring to the economy?
Yeah. I mean, this is something that to be honest, I actually really miss about living in Alaska, because fisheries there are really an integral part of the economy, and just culturally, they're a big part, both for the kind of like Caucasian community, right? But also for the Native Alaskans that live there. I mean, there's a lot of people that rely on fisheries for their livelihoods and just for cultural importance. I mean, even people that, fishing is just such a popular activity there. It's really just woven through the fabric. And so it's, to be honest, it's a little bit hard to be in California, where a lot of that has been lost, right? Because of the big declines in fish populations. And I think it does feel a little bit sad to me, because it's one thing to lose, to think about the populations declining is certainly sad on its own, but the cultural aspect and the societal aspect of having these robust fisheries and being part of the culture is also a really big loss that I don't know if often. For me, I don't see it acknowledged all that often kind of among scientists, right? We really focus on the population dynamics, or, what small things can we do, but there is this whole other societal element that goes along with it that I think is really important, and is also just as important for long-term conservation, right? Because it's really hard to conserve a resource that people don't know about, or they don't care about, or they don't have some personal connection with, right? And so I think trying to maintain that personal connection with fish and salmon here in California is also is a critical element of their conservation.
Yeah, I completely agree. So thank you so much for sharing that. Somehow we only have a few minutes left, so I only have a few more questions left for you. So let's see. We got a question asking about how COVID-19 affected your research in 2020, if it really put a halt to it, if you had to transition your time to mostly lab and writing, or how field work went, just really briefly, if
Oh man,
went.
it was a nightmare. Let's be real. It was a total nightmare, right? When when the first shelter in place happened, we had a rotary screw trap, which is like a giant metal trap up above the Los Padres Reservoir. You can't drive there. You have to take a boat. It's like a remote spot. People have to be there every day, and then a shelter in place happened. So you can imagine it was just like and then that work had to be shut down for the season, right? And for good reason. I mean, COVID-19 is deadly. I mean, it's not to be critical of that, but it was a frantic, tough time. Things have gotten better though. The university was really good about putting safety precautions in place and keeping being reasonable about it, and also getting us testing and getting us vaccinated. And so for the most part, we are back on track. We were able to keep our antennas going. We were able to tag fish this fall. So it was a slight disaster followed by, by a recovery, but that's okay. That's just how science goes. That's how field science goes.
Yeah, well, I'm happy to hear that we are on an upward swing with a more positive note, but really interesting to hear how remote some of your field sites can be and how difficult it might be to access some of your field sites. So that's really interesting too. So let's see. So this is actually my last question for you, and maybe we'll end on a little bit more of a positive note. Can you tell us what has been the most memorable part of your career so far?
Yeah, I think for me, it's actually been the travel and the opportunities to work in a lot of different places. So I've been very fortunate in that I've worked all throughout remote Alaska. I've been able to work in Greenland, in Japan, in Eastern Oregon, and now the Central Coast of California. And those places are all just so beautiful. But also, the science has given me the opportunity to, go into these streams that I would never visit on vacation, learn about fishes I would never, have a reason to learn about necessarily, and interact with people in those places. So that has definitely, I think, been a major highlight for me.
All right. Well, Haley, we have to have you back because we only touched on your work in the Carmel River. We have to have you back to talk about Japan and Alaska and all the other cool places that you have been to. So, well, somehow we are out of time. And I just can't say thank you enough for being here this morning and sharing your expertise with us and telling us all about the research that you're currently up to right now. But I have a bunch of more questions for you, and I'm sure everyone at home does too. So Haley, do you have a place online or social media or somewhere else where folks can go to learn more about you and your research?
Yeah, I have a website. It's HaleyOhms.github.io. But honestly, if you just Google Haley Ohms, it comes up. I have a unique name. You can learn more about my research there. And there's also an email address for me there. And I would be happy to take any questions or just a note to say hi. So that's the place to find me.
Awesome. Well, I will make sure to add your website address to the video description below after we wrap up this conversation here so folks can easily find it. Well, again, just thank you so much for being here. It was a huge pleasure.
My pleasure. Thank you for having me.
Of course. And of course, I need to say thank you to everyone tuning in today. And especially to UC Santa Cruz's Fisheries Collaborative Program for sponsoring today's chat. It's really supporters like you that make these online programs possible. So if you are in the position and want to help support us further, I ask that you either make a donation or become a member with the Seymour Center. You can find all that information on our website, seymourcenter.ucsc.edu, or click on the links in the video description below to learn lots more about that. So thanks again, everyone, for tuning in. Mark your calendars for the next Labside Chat on June 10th. And until then, we'll see you at the next virtual event. Thanks, everyone, and have a great rest of your day.